So I'm Lisa Meffert from Rice University. I'm an assistant professor in the Department of Ecology and Evolutionary Biology, and today we're going to talk about Mendelian genetics, so the basic laws of inheritance. Now, in the prevailing time when Mendel started his experiments, we're going to be focusing on Mendel's experiments today. They're groundbreaking experiments because at the time, the idea of inheritance was generally a blending model. So people saw that maybe a tall father and a short mother had sort of middle height children, and so they thought that there was just this sort of blending model. Now that does make a lot of sense, but what happens is that through time, and Darwin noticed this, through time then everybody would start looking the same. And he noticed that this is definitely not true. What happens is you still have variation. So when you read Origin of the Species, you'll find that Darwin was very perplexed with this blending model. He knew it was wrong, but he had nothing to offer in its place. Now he was a brilliant geneticist, but it's interesting that he didn't understand the basic mechanisms of genetics. He had no way of identifying what was wrong with the blending model. He just knew it was wrong. So what happened was Mendel came along and actually figured this problem. What he found is that there's really a particulate model, so that when a male and a female mate, each of them gives some kind of particle to the offspring, those two particles come together and determine what will be the phenotype or the trait that is expressed in the offspring. But when that offspring reproduces, then those two particles come apart again. And so in this way, you'll see that variation is maintained. So Mendel explained this perplexing issue about how variation can be maintained through time. So Mendel's insight was to do experiments. Again, Darwin was more an observational biologist, and so he couldn't, again, answer this problem in genetics. Mendel did experiments. He was playing around with these pea plants. The pea plants are particularly easy to rear because he could say he wanted this plant to mate with this plant. It was very easy to manipulate, and they had very obvious phenotypes, so traits that he could score. Things like plant height, either the plant was tall or short. What color was the pea? Was it just green or yellow? And then let's say the pea texture, was it round or wrinkled? So this was a very easy way for him to score these different traits. The first thing he did was that he made sure that he had these lines lines that through time always had the same trait. So let's say he had all tall breeding lines and all their offspring were tall, and then he had all short lines, they were all short too. So those are the parental lines, those are the pure lines. So then what he did was he would cross them. He would cross, let's say, a tall plant with a short plant, and what he found was that all the individuals would look the same. This is the F1. So what happens is you seem to have lost variation. But then what he did, he took to the next step. This is the brilliant insight he had. He crossed these individuals, so the offspring of the parental lines, with each other, and then he found you go to the F2, and then he found that the variation was maintained. In this case, 3/4 of the F2 individuals, that is in this third generation, looked like one of the parental lines, where 1/4 looked like the other. So this explained how variation is maintained in a population. And we're going to go into this in a little more detail right now. So what we're going to do is work out this particulate model of inheritance, how you cross two pure lines, get the F1 where all the offspring look like one of the parental lines. Then you cross those offspring with each other, and we'll show how you can have the variation come out again, where you can see the two different phenotypes or traits that were expressed in those the original pure lines. What we're going to use here is a Punnett square, and a Punnett square is just simply a bookkeeping tool. We're just going to figure out how we can separate out these two particles, the particle that is conferred from the father and the particle that is conferred from the mother, in producing the resultant trait that the offspring shows. So we use the term allele, which is the specific form of that hereditary particle. In this case, we just arbitrarily say that a big T, capital T, confers tallness. So when we had a pure breeding line, all individuals throughout time were tall, then we know every individual in that population has two big T's. So let's just have, let's say, a father with two big T's. So we call this homozygous. He has two of the same allele. The convention is then, so for the alternative trait, shortness, we have a small t. So this small t will confer shortness. So when we have a pure breeding line forever through time where everyone's short, we know that everyone in that population has received two small t's from each of their parents. So we're now going to take a short female and cross it with a tall male, knowing that since this female came from a pure line, it has two small t's, it's homozygous for T. So here we are, we're going to cross this tall male with a short female. First, we figure out how the male passes on his big T to all of his offspring. So remember how these particles sort of come apart. He received these two big T's from his parents, and now they're going to separate out. We're going to just arbitrarily have 4 potential offspring here. So each of the cells here represents the offspring, one of the offspring, and each cell gets a big T. Now we go to the female. The female now confers a small t to each of her offspring. Now, we have the resultant genotypes, that is the combination of alleles for each of the offspring. And we note that all of these offspring are tall. That is, if you have at least one big T, you're tall. And the term for that is dominance. If big T is dominant to little t, that is, if you have at least one big T, you're tall. So remember how we said that in the F1, everyone looked the same. Somehow you had lost the shortness. Well, what we have to do is go to the next generation. So what Mendel did was take those offspring, each of those is now heterozygous. That means they have two different alleles, big T and a little t, and cross them with each other. And we'll just go through this Punnett square again. In this case, we're going to let the male transfer either the big T to 2 of his offspring or the little t to 2 of his offspring. And then we do the same with the female. In each egg, the female can contribute either a big T or a little t. And remember that rule that we said, that if you have at least one big T, you're tall. So in this case, we have 3 offspring that have at least one big T. So we have 3 offspring are tall, and where one has 2 little t's, remember you have to have at least one big T to be tall. If you have 2 little t's, you're short. So we have a 3-to-1 ratio for 3 offspring are tall to one of the 4 offspring is short. So this is where you get that 3-to-1 ratio of the phenotype. So the phenotype is that resultant trait that you see based upon the genotype, which is the combination of alleles that you have received from your parents. So now we're going to move on to 2 traits at a time. Now, of course, remember when you were born, all the traits were determined by the alleles or the particles that you received from your parents. But we're just going to move on to 2 traits just for simplicity. And in this case, we're going to keep the big T and the little t symbols for tall and short plants. And now we're going to look at the seeds. Do you have round or wrinkled seeds? In this case, we're going to use a big R for round and a little r for wrinkled seeds. Now, he started with the pure line, so we're going to go at the point where we've got those individuals that are heterozygous for the tallness and they're heterozygous for the roundness. They are derived from pure breeding lines, so we know that each individual has at least one big T, one little t, one big R, one little r. And we're just going to use this Punnett square again in order to figure out what all possible genotypes and thus what all possible phenotypes we can get from this kind of mating. So these are all possible combinations of sperm, let's say, that this male can receive. He can have a sperm with one big T and one big R. Remember, these particles are coming apart. He can have one big T and one small r. He can have a small t and a big R. And he can also have a type of sperm that is little t, little r. We do the same with the female. We're taking this double heterozygote female and figuring out all kinds of eggs that she can produce with regard to tallness or shortness, roundness or wrinkled traits in the seed. So again, she can have a big T and a big R in an egg, a big T, a little r, a little t, a big R, or a little t and a little r. So first we'll figure out what all sperm the male can contribute when he mates with this female. So you just walk down this Punnett square. Again, this is just a bookkeeping tool, and you can see that in all 4 of these cells here, the male will be contributing a big T and a big R. Similarly, in each one of these next 4 cells, the male is contributing a big T and a little r. Again, going down the line here, he contributes a little t and a big R in this kind of sperm, and then in this case, a little t and a little r. Now we see what all the female can do. She does the same. You just fill in the spaces here. This kind of egg, when it mates with this kind of sperm, you'll see that it gets 2 big T's and 2 big R's. When this egg mates with this kind of sperm, you see you get two big T's, a little r, and a big R, and so forth. So you just fill in the symbols, just walking across, in this case, the rows, in order to figure out all those possible genotypes you can get for those two traits. Now, let's look at the phenotypes. Those were all the genotypes, all the combinations of alleles that you can get for those two traits. So now let's look at the phenotypes that you see. Remember that if you had at least one big T, you were tall. And in this case, if you had one big R, that's dominant to wrinkled seeds. So if you have at least one big R, you have round seeds. So we've used just a color coding here to say, okay, now how many individuals are tall with round seeds? So just count out the number of individuals that have at least one big T and one big R, and you'll see there are 9 possible ways to come up with tall offspring with round seeds. Okay, so we determined that there are 9 ways to make tall offspring with round seeds. So now let's look at the other kinds of combinations we can get with regard to tallness and the relative roundness or wrinkledness of the seeds. So you'll see here, we've color-coded here all those individuals who have at least one big T, remember that makes you tall, and 2 little R's. Remember, if you have one big R, you're round, so you need 2 little R's to be wrinkled. So you notice here that to have at least one big T and 2 little R's, that is, you're tall with wrinkled seeds, there are 3 ways to get this. Now let's look at the other kind of combination. What about short individuals with round seeds? Well, if you're short, then you need to have 2 little T's. And then, in order to have round seeds, you have to have at least one big R. So let's look at all kinds of combinations where you have 2 small T's and at least one big R. That means you're short with round seeds. So we have 3 ways to do that. Then finally, what do we have left? Short individuals with wrinkled seeds. Remember that you have to have 2 small T's because the big T, the tallness, is dominant to the little t for shortness, and then you also have to have 2 little R's for wrinkled seeds because one big R makes you round seeded. So you note that there's only one way for this combination of sperm and egg to get 2 small T's, 2 little R's. That means you're short with wrinkled seeds. So that's where we come up with what's called the 9:3 to 3:1 ratio. 9 ways to make individuals that have both of the dominant traits, 3 ways to make the 2 kinds of individuals that are dominant for one trait but homozygous recessive for the other, and then only 1 kind of way to make individuals that are homozygote recessive, that is, they have none of the dominant alleles. And that's how you explain Mendel's particulate model of inheritance. And the blending of Mendel's particulate model of inheritance with Darwin's insights led to the modern synthesis, which is how we see evolutionary biology today, is an important field of genetics. Thank you.